Hello student, very good morning to all of you. How are you? I hope you all are happy, healthy and fine at home. Do you remember what were we discussing yesterday? I think we were discussing about the disaccharide, and there we have seen common three saccharides are generally we have discussed. Number one is a sucrose, number second is a maltose, and number third is a lactose. These are the three common disaccharides are found in a different natural product, generally as a carbohydrate, and there we have seen as a one of the physical property which is an important property as far as carbohydrate chemistry is concerned, that is a reducing sugar or the nature of the reducing glucose. what will be the actual concept of reducing sugar? Those carbohydrates are generally, let us see here, is a disaccharide which containing as a hemiacetal group, then they exhibit or they shows or they indicate the reducing nature, means they exhibit the positive Toulon test or positive filling test. It means that there is a aldehyde group is present. Those organic compound or those carbohydrate has aldehydic formyl group property. They can show the reducing nature of that compound, means hemiacetal structure clearly shows. If you observe the structure of glucose, if you observe carefully the structure of sucrose, you will find there is a no anomeric hydroxyl group or no hemiacetal type of the group not involved in sucrose, and therefore sucrose is a non-reducing sugar. If you see or if you observe on the other hand structure of maltose and the lactose, you will find there is a hemiacetal group is present, and as we know that hemiacetal group is a potential for formyl carbonyl group, and therefore they shows positive Toulon solution as well as positive Toulon test. Similarly, the positive filling solution. These are the two tests, and how to represent that disaccharide, how to convert Fisher projection formula into Haworth projection formula with the help of some suitable example, for example sucrose, then maltose and the lactose, what kind of the glycosidic bonds are formed or glycosidic linkages are present there. That also we have learned with the help of their structure according to as a Haworth projection formula. So now what we are going to discuss today, right now at present, is look at here, we are discussing about the synthesis of glycosides. Glycosides means formation of the glycosidic linkage when polysaccharide. Today we are going to discuss about the polysaccharides, that is a polysaccharide, that is the point of discussion. Moment you think about the word poly clearly indicate as many, large number of these type of the thing whenever there is a polysaccharide, and a saccharide means that carbohydrate, as we know that glucose unit. Polysaccharides means number of or large number of monosaccharide units are linked together with the help of glycosidic linkage is known as a polysaccharide. Let me tell you once again, polysaccharides are formed by linking large number of monosaccharide units by glycosidic linkage. Glycosidic linkage is the word which is highly important. Glycosidic linkage is found here. Glycosidic linkage, or we can call it as a bond, glycosidic bond. Linkage or bond, one and the same thing. These are the synonym word for that linkage. Starch, cellulose and glycogen. Starch, cellulose and glycogen. These are the three word generally. Starch, cellulose and glycogen, the most common natural polysaccharides. Starch is a storage carbohydrate of plant, an important nutrient for humans and other animals. Cellulose is the main constituent of cell wall of plant and bacterial cells, and it is also main constituent of wood and cotton. Glycogen constitutes storage carbohydrate of animals and is present in liver, muscle and brain. It is also found in yeast and fungi. Let us see here some of the structure of that starch, how the starch are present in that. Let us see here one structure I am drawing here. This one O, then this one once again. Here I am drawing this structure, and I will explain later on why I am showing this type of the structure. As we know that glucose, how the glucose is formed, that we are going to learn first. Let me draw some structure and later on we will discuss about that one. Here the hydroxy group is generally shown below the plane, here above the plane, and here, as we know that generally here, let me make the correction over here. Here oxygen atom, and this type of the linkage is found over there, and where it is found CH to OH, then same you can write OH, then OH here, then CH to OH. Similarly one OH here, and here CH to OH. This is a structure of what we can call it as a alpha beta. This 1 2 3, and one more structure is drawn over here. So the structures are this one generally is written here again, once again CH2OH, here OH and OH. This type of the linkage, what do you think? This linkage is generally, look at here, below side is the beta side, here also as a beta. So beta side at 1 and 4 position, as we know this is 1 2 3 4 5 and 6. This is the framework of that D-glucose, and this is alpha D-glucose. Therefore you can call it as alpha connected via 1 alpha dash 1 carbon atom and 4 carbon, 1 2 3 4 5 and 6 at 4 carbon atom. So we can say alpha 1 4, this is linkage, alpha 1 4 glycosidic linkage, alpha 1 4 glycosidic glycosidic linkage. This is a linkage found in the figure. This figure indicates that this is the figure of amylose. This is a figure of amylose. Amylose, it is one of the starch amylose, and second structure I am going to draw over here. So that structure is, let us see that structure is somewhat this one. Let me draw that structure over here once again. O, not O, here O. Then I am trying to explain this structure O. Then here CH2, and then it will be attached with that oxygen atom. Then this one, then we can draw O here, then this one, then O, this one, two structure over there or one, and this side also we can draw, not this side, again goes below oxygen atom, then below, then we can draw over here this type of the structure. Then similarity, look at here again, once again hydroxy group below the plane and another hydroxy group above the plane. Here we can write CH2OH. Here also has the same hydroxy group below the plane, one of the hydroxy group above the plane, then you can write CH2OH. Similarly, in those structure also, here you can write OH and OH here. Here you can write CH2OH. Here also same, one hydroxy group below the plane, one hydroxy group above the plane, and now here one hydroxy group below the plane, one hydroxy group above the plane, and then CH2OH. Then same thing happening over there. Now look at here, this is the carbon number 1, 2, 3, 4, 5 and 6. Here again carbon number 1, 2, 3, 4, 5 and this 6. Now look at this one is alpha joined through 1 and 6 glycosidic bond. This one 1, 6 glycosidic bond, and there is a branch. This is a branch, right? This is a branch present in this structure. And now if you look at, this is similar to as this one, so there will be again alpha 1,4 glycosidic linkage, right? And the figure of this figure is for amylopectin. This is a figure for amylopectin. This is a figure for amylopectin. Amylose and the amylopectin is, there are two components present in the starch. This amylose, which is soluble in water and form as a blue colored complex with iodine, whereas amylopectin is insoluble in water and form as a blue or green or violet color complex with the amylopectin. So that is these two components, both the component in starch. As we know that in the starch two components are present. Number one is amylose and second one will be the again is amylopectin. Amylose, amylopectin. Amylose and the amylopectin, these are the two components present in the starch. This is all about that. Let us see one. First one, we are going to describe as a starch. You can designate, you can give the point is a A over here. Now here you before that use your brain power. How do you use? Is galactose and aldose are ketohexose? Have you seen the structure of galactose? Yes, galactose and D-glucose are what we can call it, they are epimers of each other. Epimers means what? The optical isomer having same molecular formula as well as structural formula also same. The connectivity of carbon atom also same, but they differ. The configuration at only one carbon atom. So that type of the phenomenon is known as a epimers, and therefore galactose is a aldohexose. Which carbon in galactose has a different configuration compared to glucose? You know that, right? Which carbon in galactose has a different configuration compared to glucose? If you compare two structures, one is a D-glucose and another one is a D-mannose, they are also as epimers of each other, but they differ in a configuration. At what carbon atom, at what chiral center? They are differ at only one, at C2 carbon atom. Fermyl group CH2, if you imagine the structure of D-glucose and D-mannose according to as a Fischer projection formula, top we know that fermyl carbon carrying as carbon number 1, and then later one is the second. At a second, that hydroxy group which is present at right hand side in the D-glucose, whereas in D-mannose that hydroxy group are present on the left hand side, and therefore the optical rotation or absolute configuration of these two are different. Therefore D-glucose and D-mannose are epimers of each other. Second best example, one is a galactose and another one is a D-glucose. D-glucose, as we know that hydroxy group are present at everything is the same, but the configuration is a change at carbon number 4. If you observe carefully at carbon number 4 starting from beginning, so lowest chiral carbon, after that there is a carbon atom which has in the D-glucose the hydroxy group which is present on the right hand side, but if you see in the galactose it is present on the left hand side, and therefore their absolute configuration are different at C4. So that type of the thing you should remember. Draw the Haworth formula of alpha D galactose and beta D galactose. As we know that Haworth formula, you first write down or you draw the structure according to as a Fischer projection formula, then convert into as a Haworth projection formula. It's a very simple. Those group which are lying on the right hand side, they goes below the plane, and those group which are lying on the left hand side, they should goes above the plane. That the trick, that the rule you must remember while converting Fischer projection formula into Haworth projection formula, and it's a very easy. Then which disaccharide among sucrose, maltose and lactose is or are expected to give positive filling test? As we know that disaccharide means two monosaccharides are joined together. That linkage we know that glycosidic linkage or glycosidic bond, and after that observe that structure carefully. If hemiacetal structures are present, they are they are taint, they are actually shows positive feeling test. So that is the general tendency of that, and therefore except sucrose other two, maltose and lactose, are reducing sugar because they shows positive feeling test, whereas in a sucrose there is no hemiacetal group and therefore it does not show the positive feeling test. So that's the reason. Got it? Okay. What are the expected product of hydrolysis of lactose? As we know that lactose is present in the milk. Remember, everyone, remember, we are using in day-to-day life, in a day-to-day at the morning when we are wake upping, and after cleaning our teeth or our mouth, when we fresh, we are drinking some tea, or what we can call it as some of them are drinking as some of they have the milk, because that milk containing as a lactose, and the lactose is made up of by one is a galactose and another one is a beta glucose, and therefore it is made up of by the two monosaccharide unit. One is a galactose and another one is a D-glucose. So that's you remember. And after the hydrolysis, hydrolysis is carried out generally in presence of acid or either specific enzyme, generally as a invertase. It is a specific enzyme used in a hydrolysis, and after the hydrolysis, that lactose is a disaccharide, and disaccharide get converted into the monosaccharides. One is a galactose and another one is a beta glucose. We know that. Can you think, when you chew plain bread, chapati or bhakri for long time, it tastes sweet. What could be the reason? So that reason, that type of questions answer you find out and then you tell me later on. But let us see here. First one is a starch. What do you mean by the starch? At the beginning I already told you that starch is a polysaccharide. Starch is a polymer of alpha D glucose. Look at here. That starch is a polymer of alpha D glucose. Here alpha in a amylose, only alpha linkage is found. Here in a branch there is alpha 1,6 glycosidic bond or glycosidic linkage, and here alpha 1,4 glycosidic linkage. Means in the starch is a polymer means a number of monosaccharide units are connected together with glycosidic linkage, and that's why it is called as a starch is a polymer of alpha D glucose. Starch has two components, namely, I already told you that starch has two components, namely amylose 15 to 20% and amylopectin 80 to 85%, and amylose is soluble in water and forms blue colored complex with iodine. That is a speciality of that amylose. It is soluble in water and it forms the complex colored with that iodine, and it contains 2 to 1000 alpha glucose unit linked by alpha 1,4 glycosidic linkage, giving rise to unbranched chain of variable length. And amylopectin, second component, amylopectin is another amylopectin is a water insoluble component of starch which forms blue violet colored complex with iodine. It is a branched chain polysaccharides, and in amylopectin, chains are formed by alpha 1,4 glycosidic linkages between alpha glucose unit, whereas branches are formed by alpha 1,6 glycosidic linkage. This is all about the glycosidic linkages. Now second one is a cellulose. Let us see here the structure of that cellulose, how the cellulose, what kind of the linkage is present in cellulose, that we are going to learn here. Let's see what's it. So here generally what is going to happen, see over here cellulose. So here how the cellulose, what type of the bonds are present over there. Here oxygen, then I am drawing this structure once again here, this one oxygen atom, and then I am drawing these structures once again here like this. This is oxygen, and then here this type of the structure I have drawn over here. Now look at, this is anomeric carbon atom, and that hydroxy should be on a beta side. Therefore this is a structure according to as a Howard projection formula. It will be 1, then this is a 2, 3, 4, 5, and here CH2OH 6. 1, 2, 3, 4, 5, and this will be 6. Same here hydroxy group, as you know that D glucose has structure. This one hydroxy group is over here. Same thing here, hydroxy group below the plane, this hydroxy group I am putting above the plane, and here CH2OH. In the same way you can write here hydroxy group below the plane, and one of the hydroxy group should be above the plane, then CH2 and OH like this. So this is the structure, and what type of the glycosidic linkage found in cellulose. This is a figure of cellulose. This is a figure of cellulose, right? This is a figure of cellulose. What type of the bond is taking place over there? Beta 1, this is 1, and this is a fourth one, beta 1,4 glycosidic bond. Glycosidic bond or linkage, glycosidic linkage. This is the beta 1,4 glycosidic linkage is found in the cellulose. Now let us look at some description about that cellulose. Cellulose is a straight chain polysaccharide of beta glucose unit linked by beta 1,4 glycosidic bonds. Chemical hydrolysis of cellulose requires use of concentrated strong acids at alpha at high temperature and pressure. This implies that the beta 1,4 glycosidic bond is very strong and difficult to hydrolyze. Human do not have enzyme which can hydrolyze this linkage. Hence, cellulose cannot be digested by human beings, and it serves as the fibrous content of food useful for bowel movement. So this is all about that one. Now third one, C is a glycogen. Then this we talked about cellulose. Cellulose, if you see the structure of cellulose, you will find here, this is the structure of cellulose. There is a beta 1,4 glycosidic linkage is present, and this type of the enzyme that is not present in the human being, and therefore human being cannot digest this cellulose, and therefore it is useful. It serves as the fibrous content of food, useful for bowel movement. So that is about that. Then last one will be the C is a glycogen. Is a glycogen. And when you talk about that glycogen, so glycogen has its structure similar to that of amylopectin, but is more highly branched. That is synthesis of glycosides, glycosyl donor acceptor concept. What it will be, that glycogen has its structure similar to that of amylopectin, but it is more highly branched. That we have seen amylopectin structure, we have seen how alpha beta 1,6 glycosidic linkage is present and alpha 1,4 glycosidic branch, but this is a very simple branch over there. But in a glycogen there are highly branched structures present over there. So in this way we have seen there are three type of the polysaccharide. One is the starch, second one is the cellulose, and third one is the glycogen, where they are found and how they are found, what type of the glycosidic bonds are present over there. So this is all about the preliminary introductory, or what we can call it as a basic introduction we have seen regarding that polysaccharides. But tomorrow onward we are actually going to start the synthesis of glycosides, that is a glycosyl donor acceptor concept. What is actually it means, how that anomeric hydroxyl group is important while forming, how it becomes as a electron donor or acceptor, in what condition, at an whether it is in a acidic condition or basic condition, how the reactions are taking place, how it is very very important as a research point of view. And therefore from tomorrow onward we are going to start the actual synthesis of glycosides. Whatever we have learned so far, this is all are the primary level information we have seen. We have seen general introduction regarding that polysaccharide means what type of the common polysaccharides we can study. Then we have seen disaccharide and what type of the disaccharide, what kind of the bond they can form, what type of the glycosidic, and then we have seen reducing property, whether they are non-reducing sugar or reducing sugar. That type of the concept we have seen today here, but tomorrow onward is very very important point we are going to discuss, that is a synthesis of glycosides, that is a glycosyl donor concept. So that we will see in the next lecture. I think with this let us stop today here, remaining we will see in the next lecture. Thank you, have a nice day.